So the last part of lecture 8, part 1, is chemical methods for controlling microbes, like how we kill them using chemicals. And this is going to be very familiar to a lot of people because we've used things like bleach and soap and different disinfectant chemicals like Lysol. And depending on where you work, you may well have used some of these different things. I'm not that interested in the different specific products because in each case you've got to read the instructions and just follow the guidelines. I want you to understand why the guidelines exist. And so, again, I want you to know the different methods, what they're used for, when they're not used. But the thing I want everyone to remember after taking this class is why we have to use them in a certain way and how if you use them wrong they won't work. And that comes down to really the concepts we've seen before in this lecture, but here when we're talking about chemical disinfection we use what's called the 3 C's: chemical, concentration, and contact time. So which chemical do you use? Is it appropriate? Is it going to damage the surface you're trying to disinfect? Is it going to work against the type of microorganism you're trying to get rid of? Is it concentrated enough? Is it too concentrated? Something like ethanol has to be around 70% ethanol with 30% water to work. That's all I'm going to say about that. And then contact time. This goes back to the decimal reduction time and how a lot of people think that disinfectants work instantaneously, but they don't. So you may well have rules where you work that say soak this thing for 10 minutes, and that is a important rule to follow. So the first of the methods we look at would be like phenol. This is what Joseph Lister used. He sprayed on surgical wounds, and that would have been the most painful thing a patient ever experienced because phenol is extremely irritating and it burns, but it also kills microorganisms. So there's that. We have more modern relatives of it that are less irritating, and we can use them on skin. This is one that's used in clinics more, so some of you may be more familiar with it, but it's not a sterilant. It can't kill spores in any meaningful timescale. Halogens I want to talk about a little bit more. This is a big group, and it's the most widely used disinfectants in the world. It includes things like bleach and iodine and chlorine gas. And these are all capable, hypothetically, of sterilizing samples, but that's not practical because it takes too long. Sort of like boiling water. Boiling water can sterilize anything, but it might take, 2 days of boiling, and no one is going to have that kind of time. So one example is iodine. Often this is used for things like disinfecting skin. Bleach is a very common disinfectant. And it's damaging to skin. You wouldn't use bleach on skin, but you would use it on a floor or a countertop. And bleach is not expensive, and it is kind of the gold standard that microbiologists use to disinfect surfaces when we need to. The other gold standard is fire. If you really want to disinfect a surface, you throw ethanol on it and set it on fire. But that's not feasible in a house. It is feasible in a lab where every surface is made of stainless steel and fire won't damage it. But yeah, don't do that at home. Bleach is great. Alcohols you'd be familiar with from the hand sanitizer that everybody was fighting over in March 2020. And what alcohols do is disrupt plasma membranes, or they disrupt lipid bilayers. And in order to do more than that and destroy proteins, they have to be hydrated. And so 70% ethanol is the best concentration. Ethanol is also used to disinfect surfaces. And in some cases, is thought to be able to sterilize surfaces, but not through poisoning the bacteria. It's because we use it to mechanically remove them. It can separate bacteria from surfaces, so we can use it to physically remove them by wiping them away, but it doesn't kill them. So we can remove endospores using alcohol, we just can't destroy them using alcohol. Heavy metals is the next one. Not much information about this beyond what's on the slide. Some others you can look at. Surfactants, some of these are great, like the quaternary amines. These disrupt cell membranes, and some of these can be used in a lot of jobs students might have had. You would have used a quaternary amine. Some of the weirder things would be gases. Most people wouldn't have been exposed to these directly, but if you've worked in a clinic, you've probably used tools that were sterilized with ethylene oxide. And this is a gas that reacts. It's extremely reactive. If you see the structure of this and you know organic chemistry, you look at it and say, how does that molecule even exist? It shouldn't, and it can't when it touches other things. It reacts with them, so it kills cells, it can kill spores. And so if you have like complex medical equipment with a lot of tubing and you need every little bit of it to get sterilized, you can put it in an ethylene oxide chamber. Chlorine gas is another sterilant, but it's extremely dangerous. It's extremely dangerous. So is ethylene oxide. But chlorine gas is more common. We use it to do things like disinfect drinking water. So if you go to a water treatment plant where they make drinking water, they put chlorine gas into it, and that will usually won't sterilize it, but it will disinfect it. And if the tap water tastes a little bit like chlorine, that's a good thing, because that means there was some disinfectant still in the water by the time it got to you, and that means your pipes are likely to still be safe. If you don't want to drink water that tastes like chlorine, you can use a charcoal filter to get rid of it, or just set water out on the counter. And chlorine is a gas. It will eventually evaporate from the water. It doesn't take very long. Like, one thing you might consider is, what do you do if your office building is contaminated with anthrax endospores? This happened in the U.S. Senate office building and other buildings in Washington, D.C. in 2001, and various different liquids and gases were used to clean the office buildings. I mean, think about, how are you going to clean the inside of a computer? A computer has a fan, and it may have ingested some of the anthrax spores. And sometime in the future, it might blow them out again. And so, how do you kill anthrax spores inside a computer without destroying the computer? And in that case, you would need a gas. And so that's what they had to do, use various gases. They wouldn't use chlorine gas for that, but they did have others that they used. Oh yeah, hydrogen peroxide, this is an oxidizer. So it works kind of the same way that bleach and chlorine gas do. They oxidize everything it touches. And so that's going to add, it's just going to change the chemistry of every protein and everything else. So it's useful for that. And then the last part of this first set of slides is this phrase, kills 99% of harmful microorganisms. I've seen this and I hate this. It doesn't mean anything. So I want to walk through that with you. What is 99% killing? There are two different ways I could interpret this. One is that it reduces the population by 99.9%. So it's 3 tenfold reductions. If you start with 100% alive and you go to 90, that's killing 10%. If you kill 99%, that's a 100-fold reduction, going from 100% down to 1%. If you kill 99.9%, you're going from 100% down to 0.1, so that's a 1,000-fold reduction. Okay, great, but what if your initial population was 10 million cells? Also, does it kill endospores? Does it kill endospores? What is the population? I assume this is what they're talking about when they put a number like this on it. But they might be talking about microbial species. They might be talking about how it kills 99.9% of microbial species. In that case, what does kills mean? Does it kill them quickly? And 99.9% of harmful microbial species doesn't mean really anything. There are only about 100 pathogenic bacterial species. So there really aren't that many. So this would be, it kills 999 out of 1,000. So it's probably talking about this, and it's just not that great. So anyway, the point is, if you see this phrase, ignore it. Yeah, it reminds me of the Consumer's Digest thing. Whoops. Hello. Hi. It reminds me of the Consumer's Digest thing where you'll see on car commercials, they'll say that a car is a Consumer's Digest Best Buy. And what they want is for you to mistake that for Consumer Reports. Consumer Reports is a magazine where they do extensive product comparisons, but they won't let anybody quote them in advertisements. So this company, Consumer's Digest, just exists to sell the ability to say Consumer's Digest Best Buy. So if you ever hear a product advertised as being a Consumer's Digest Best Buy, you should get angry that they are essentially trying to mislead you in their advertising. And that's how I feel when I see this phrase. That's it for the first part of this lecture. The second part will be about antibiotics, so remember to check that out. Take your time with these. These are important concepts. That's all there is to it. They're important things to know. I'm, again, much more interested that you understand the conceptual stuff than individual different chemicals. So there might be one or two questions comparing the individual chemicals, but definitely questions on things like the kinetics and why one works versus another. So check that stuff out. And I will see you at the next video. That is the next playlist.